Yeah, is my screen is visible?
No, ma'am.
Now?
Yes, it's visible.
Okay. So, yesterday we were discussing about this tree and the options which we are getting, right? So, basically, here in this tree, we were discussing some options related, like for the editing purpose and all, like how we can edit, what is the meaning of distance scale, divergent time, then time scales, ancestors. So, let's discuss or let's revise it one by one. But before that, is there any doubt? Okay, so here, yesterday I have discussed about this different layouts. Like if you want to give some different tree styles, you want to try that, increase the width, increase the height, you can just use the layout. Then here, branch length. So, what is a branch length? Can somebody please explain? We have already discussed this yesterday.
Branch length corresponds or it represents or shows the evolution between the difference in evolution or the mutations happened between two species.
Hmm. So branch length, you can say it's a measure of evolutionary distance, which is typically expressed in the terms of number of substitution per site. Means here, for the nucleotide sequence, the branch lengths are often measured in the number of nucleotide substitution per site. Means, example, if we are getting a zero point 0.1, so it's a zero point 0.1 substitution per site there. So here, if you want to increase the font size of a branch length, then placement, you want to give above the branch, below the branch, or automatic. Then precision, how much decimal points you want. Then hide, if you want, if any length is shorter than a specific length, if you want to hide that, you can use this option. Then we discuss the frequency info or a statistical info, statistics info also. So basically, in the frequency info or a statistics info, here we are getting the bootstrap, which is supporting to the values that are present and indicating the proportion of the bootstrap replicates, like a resampling of the original data set. So if you are having a 10 times, 20 times, or 30, so it means like a 10 times the branch or clade was supported in the 10% of the bootstrap. So this is what it is showing you actually the percentage about the statistics. Uh, then yeah, then here if you will go down, the another parameter we are having is a distance scale. So what is a distance scale?
It measures the genetic divergence between taxa.
Hmm. So actually, in the context of the phylogenetic tree and the evolutionary analysis, a distance scale is referring the measurement of evolutionary divergence or the genetic distance between the sequences, taxa, or a species. So if the distance scale of 1.01, if we are getting in the molecular phylogenetics, it is indicating the moderate level of the genetic divergence with an average of 1.01 substitution per site. So it is providing the quantitative insights into the evolutionary relationship. So if there is a higher diversity, this value, the distance scale value will increase. But as of now, here in our data, like all these sequences are very closely related. They are from a single species only. So that's why you're not getting the higher distance scale. But yesterday, in the Pushpit's systems, discussed about, I show you about the distance scales, right? Then next you are getting here the ancestors, like a divergent time, how much time, sorry, this is what the diversion time, then time scale, it's not applicable for this. Then you are getting about the ancestor. So in the mega, ancestor is referring to the ancestral sequence, or you can say the node in the phylogenetic tree, which is representing the hypothetical sequence at a point of divergence in the evolutionary history. So these ancestors are critical for understanding the evolutionary relationship and the dynamic of molecular sequences over the time. So this is what, like, if you want to see all the ancestors, you can give show all, then show most probable, then hide the ambiguous, and none if you don't want to display the ancestors there. So this is what you are getting the original tree, and here you are getting the bootstrap consensus tree. So bootstrap consensus tree, it's a type of phylogenetic tree which is showing you the information from multiple bootstrap replicates. Like here you are having the values. Yeah. So here we are having the values. These are nothing but the number of bootstrap replicates. As of now, we have given the bootstrap replicate 50 in the last session. So out of that, 20 times it is there, 2 times is there. So highest the bootstrap value indicating the highest confidence, highest replications about it. So how to interpret it? So if the number which is associated branch, like example, if you are getting like a 70 or 80, it is indicating the branch was supported by that percentage of the bootstrap replicates. So higher you can say more than 70, lower you can say it's less than 50. But as of now, we have given the 50 cutoff. So you can just change the cutoff accordingly. But in real time, as I have told you, that we need to give the 100 minimum value about it, right? So this is how basically we can interpret the tree and we can generate using the mega. Do you have any doubt here?
Ma'am, can you tell me about the site coverage part? What is site coverage?
There we are having the site coverage.
Ma'am, next to the frequency one, statistics frequency info.
Yeah, yesterday actually when we clicked on the site coverage, it is displaying the percentage only. Like how much site it is covered, that percentage it was like it is displaying the percentage there instead of the bootstrap values.
Ma'am, percentage of what?
Percentage of bootstraps there means it is showing you like if you are getting a 10% or a 20%, so it's showing you the 10% replicates of bootstrap. So bootstrap value it is showing about the site coverage.
Okay, ma'am, got it.
Yeah. Hmm. So this is about the phylogenetic tree. I have given one exercise question. Some no, I didn't give that. Just a second, I will upload. Uh, So basically, yesterday I am giving the questions to you, but our team said that MCQs, like multiple choice questions we can give as a test on Saturday. So what I did, mm, like I I want to give some practical questions. Is it fine, like, if I will give some practical question based on the UCSC database, because that database we missed some assessment, which I want to give you right now because tomorrow we have a holiday. So you can try that questions. Is it fine? And MCQs will be provided on Saturdays.
Yeah, ma'am.
Yeah. Okay, I have given one practice example.fasta file. This is a file for the nucleotide sequence. So can you construct the phylogenetic tree using that file? And like you can just generate it by tomorrow, not an issue. On Friday, you can show me. No, actually on Friday I'm not there because there is one tool you are going to discuss on Friday called PyMol, which is going to take by the another trainer who is expert in CAD. But on Monday I will join again for the Linux, so you can show me on Monday. There's enough time you're having to solve the exercise question for the UCSC and to generate the phylogenetic tree for this practice example FASTA file. Is it fine?
Yeah, ma'am.
Hmm. Okay, I will give you the question at the end of the session. Now we need to be install the Linux. Okay,
Excuse me, ma'am.
so yeah,
But can you also upload those PPTs where you have these question examples?
sure,
Because PPTs are not there.
sure. I will. Yeah, most of the PPTs of the databases I have uploaded. I
Yeah,
think
no, these
I didn't
alignment.
upload it about the blast and all.
Yes, yes.
Yeah, okay, I will upload it. Sure.
Thank you.
Okay. Uh, Next thing actually we are going to start is the Linux. Okay. So I just want to know, like, whoever is having a Windows system and whoever is having a Mac system, like, is there anyone who is having a Mac system here?
I think Yash works on Mac system. I mean, I'm not sure, but I think he works on Mac.
Okay. Fine. So I think he's not uh, in the session right now. Not an issue because what I want to say is about the Mac. If you are using a Mac, you don't need to be install the Linux. Uh, and there is some another way of installation of tool. So if possible, you can arrange one Windows system. Otherwise, we can proceed for the Mac. But in a Mac, we are facing some problem for installation of the tools, and some tools are not supporting at any cost in the Mac. So at that time, then we need to provide the input file and then you can proceed for the pipeline. So better if you have already the Linux system, otherwise in Windows, we can install the Linux. Okay. So as of now, he is not here, no problem. I will tell him later about it. So let's start some Linux introduction first of all, because before installation, you should know what is Linux and why it is important for us. So, can anybody tell me what is Linux?
Ma'am, it's a operating system similar to Windows or Mac, and
Mhm.
we have quite an independence. Like we can install various software and that all stuff.
Right.
Like
I think it's a command-based, free, uh, uh,
Mm-hmm.
malab, I mean, operating system which is freely
Mm-hmm.
available.
Yes. Right. So, Linux is what? It's one of the most powerful, flexible family of operating system, which is free to use and share. Means what? This Linux is a open source operating system. You don't need to buy it. You can use it. You can install it, or you can get it for free, and you can share also. And that is completely free. It was created by a person named Linus Torvalds in the 1991, and that's what by his name The operating system is name is Linux. Uh, like whenever we are saying about the Windows, if you know, like there are different types of Windows we are having, right? Windows 7, Windows 8, 11, 10. Likewise, for the Linux is also we are having a different types and different versions of it. So, if I will give you some example, like a Debian is there, Fedora is there, Linux Mint is there, Red Hat is there, Ubuntu, Solus, then Kali Linux is also there. So these are some different types of the operating system we are having. Just give me a 1 second. Okay, so now these are the different types of Linux we are having basically. Out of that, we are going to be focused on Ubuntu. Okay, we are going to be use the Ubuntu further. Now, first and foremost thing we need to be understand like why Linux, why we are not going with the Windows or a Mac. So actually the main advantage of the Linux, it's a open source operating system, just like a Microsoft or a like Windows. You don't need to be buy that. You can install it freely. So that's what we can say. It's an open source operating system. It's a secured OS. Like you don't need to be install the antivirus for that. It's a very secured operating system. The data in the Linux is very secure. Then it is compatible for the large amount of data, because as of now we are dealing with the sequences, we are dealing with the so much biological data, and nowadays we are getting a very huge biological data. So how to analyze that data, because manually it is not possible to handle that data. And in Linux we are having so many powerful commands like grep, head, tell, sed command is there, then tr command is there. So with the help of so many powerful commands, we can handle the data very easily. It's a command line based operating system. Most of the things we can perform with the help of commands. I'm not saying like that we cannot perform this in any another system like Mac or uh, let's leave about the Mac, like a, because Mac is also same kind of Linux. You can perform all the commands in the Mac like a Linux. But I'm not saying it's not we cannot perform in a Windows, but some commands, because if you want to handle a huge amount of data, let's consider you are having one FASTA file with a 10 GB file size, and you want to count like how many number of FASTA sequences are there in the file. So, if you are going to open it manually, if due to the huge file size, it is not going to open anyhow. And maybe let's consider you try to open it, it's open. But due to the large number of sequences, you are not able to count exactly how many sequences are there, because it's not feasible, it's not possible. So in Linux, we are having a grep command for that, which is used for the matching regular expression. So with the regular expression matching, we can find out like what kind of or how many number of sequences are there. This is just one example I'm giving. How can we handle a large amount of data? But there are so many things which we can't perform manually there. So that's why with the help of due to the commands, we can handle the large amount of data. And the main part here, why Linux is important, because most of the tools are built in the Linux environment and they are compatible in the Linux environment. Like whenever you are going to perform a CAD at that time, you can use Windows because that softwares they are compatible in all the operating systems. But if you want to perform specifically the NGS data analysis, those applications are not compatible in Windows. You have to run the pipeline or the tools in the Linux operating system only. Is it clear?
Yes, ma'am.
Yeah. Now, How to install the Linux? That's the main thing because we are already having the Windows. So how can we install the Linux? So basically, there are two ways, or we can say three ways to install the Linux. You can install the Linux by VirtualBox. Okay, by VirtualBox. VirtualBox is what? Do you have any idea?
No, ma'am.
So VirtualBox actually it's one of the software uh, which is you can say VirtualBox is what? It's one of the software which is creating the virtual environment, which is creating the virtual environment in your Windows operating system. So inside that virtual environment, we are installing the uh, Linux operating system. Means what? We are having a Windows. Inside that, we are creating one virtual environment, and in that virtual environment, you can install any operating system. So that's what we are going to install the Linux inside it. It's one of the software actually. So this virtual box, like if you want to install, you require some configuration. Minimum 4 GB RAM. Minimum, I'm saying, minimum 4 GB RAM you required. Then you required 100 GB of HDD at least, 100 GB of space in any one of the hard disk drive, and yeah, like this is what some configurations you required. And yeah, 64-bit operating system. Right now, we are not having some 34-bit. Most of the systems are 64, but still, this is the requirement. But like in the virtual box when we are installed, the drawback is like the space is very limited now. Means if you provide the 100 GB, if the data is exceeding more than 100 GB, at that time it is not going to work even if you have a space in Windows. Still it is not going to be allocate, like you cannot allocate that space after the installation of VirtualBox. Then again you have to uninstall it, allocate the more space, and then you have to install again everything. That's not feasible, correct? So that's what the second installation actually we are going to do. That's a Windows subsystem for Linux, WSL. So in the WSL, we are installing it, or WSL or Ubuntu LTS, you can say. So Ubuntu LTS or a Windows subsystem for Linux, it is going to be install the Linux operating system inside the Windows. It just like in a Windows we are having the command prompt, and in the Linux we are having the terminal. So it is opening the Linux terminal. But you at that time you are not allocating a specific space to that WSL. It is going to be take a space from the Windows system only. Is it clear?
Yes.
Yeah, ma'am.
Like, if you have any doubts, you can tell me. I can repeat it. Means, see, for the virtual box, if you install, if you give before installation, you have to give the space for that. If you give the 100 GB, and maybe if your data is like 105 GB, so at that time you are not able to run. Okay. But if you install the LTS or a WSL, at that time, maybe let's consider if my drive is having a space like let's see, this E drive, I'm having a space 141 free now. So I can use all this 141 GB space if I'm opening the terminal or if I'm running the command from this drive. I'm not allocating a specific space. Whatever the space is available here in the Windows, we can use that. Here also, I can, if I'm running this from this drive, I can use all the 229 GB free space. I'm not allocating a specific 100 GB or 50 GB for it. Is it clear?
Yeah, ma'am, got it.
Hmm. Okay. So this is what the second way of installation, and the third one. So third one is what? It's a single boot or a dual boot. Do you know what is a single boot or dual boot?
Ma'am, single boot is only having one operating system in your laptop or computer. But
Mhm.
dual boot, where your system is divided into two different operating systems, Windows as well as Linux.
Yes, exactly. Exactly. So single boot is what, like, this is what our system right now. Our system is only having a one operating system, right? Our system is only having a one operating system. Right now we are having a Windows. So that kind of system is only having the Linux. They are not having the Windows there. So that is what a single boot. But dual boot means what? Like, if you have a two operating system, so whenever you are starting your computer at that or your laptop, it is asking to which operating system you want to enter, or to which operating system you want to open. So you can select and that operating system will open. So this is what a single boot and dual boot. So for the dual boot installation, you require some Ubuntu files, like you need some installation file. So that's actually the like you have to install it by using some drives and some pen drives and all, some external drives and all. So that's what, like, this is not a feasible for us right now, so that's what we are going with the second type of installation, WSL. Okay, is it clear?
Yes, ma'am.
Hmm. So now to install the Ubuntu or WSL, you require some options. You required some options should be checked in your system first. So here, if you will see that, you have to go with the control panel, and you have to open turn Windows feature on or off, application using start menu, then checkbox next to virtual machine platform Windows subsystem for Linux. So just follow what I'm doing right now. You have to go with the control panel.
Ma'am, I have the Ubuntu app on Windows. So is the same?
If what is the version of Ubuntu?
I'll check about it.
Yeah, we required Ubuntu 20.04
Okay, ma'am, I'll check.
or more updated than that, not the like lesser version. But make
Oh,
sure if you have 20, it will be better because some tools are not compatible in higher version also.
okay, ma'am.
Hmm.
Ma'am, it's not showing version. I'll check it. I'll let you.
Yeah. Okay, so here we have to go with the programs. Then you have to go with the turn Windows feature on or off. So please go through here.
Ma'am, it's 20.04. Will that work?
20 point.
04.
Yeah,
20.04.1, right?
yeah,
So
that's what we want. Huh?
take.
Ma'am, what next?
Hmm. Once this is done, now what you have to do is if you just scroll it down, some option you should be checked in your system. Out of that, we are having the virtual machine platform. Here you have to check. Then you have to check the Windows hypervisor platform. And then the third one is Windows subsystem for Linux. So these three option you should be checked if it is not.
And ma'am, what about others? Like in mine, many are checked. So what should I do?
It's
Should
fine.
I?
It's already checked if it is, so that's fine. But for virtual, so for this Linux, we require this three.
Okay, ma'am.
Hmm. Now, once you will click on OK, just a second. I need to see if any another option we required or not. One is a virtual machine hypervisor.
Ma'am, it's showing.
Then when what?
Uh, ma'am, it's showing that I need to restart.
Yes, that's what I'm saying right now. Like once you clicked on OK, it will ask you to restart your system and you need to restart.
So do I restart it, ma'am?
Yes, you need to restart, but just wait for a minute. What we can do, because we just have a 20 minutes, I will share the questions and because that's the requirement actually I want to set for the Linux so that we can install the Linux directly, right? So can I give the questions now then, so that maybe you can just copy paste the question somewhere and you can restart your system, so no need to join again then.
Okay, ma'am.
Is it fine?
Yeah, ma'am.
Yes.
Yeah, for everyone I'm just telling this thing. So just uh, let me give the questions. I can give in the chat box. Can I give in the chat box, or can I write in the Word document so that you can also? Okay, I will give in the chat box. It will be better. So what you have to do is you have to retrieve all genes from Homo sapien, from human, along with transcript, exon, CDS, strand information from UCSC. Uh, Previous, like on the session we discussed for the chromosome 1, but this is not for the chromosome 1, this is for the whole genome. You have to check this, okay? You have to retrieve all the genes for human along with the transcript, exon, CDS, and the strand information from UCSC database. This is your first question. Okay.
Ma'am, it will
Second
be a
question.
very big. Uh,
No, you have to do it in Excel file, so it will be not good. Okay. You require the Excel file how we did for the
yeah.
first exercise question there. So no, it's not very big file. I'm not
Okay.
asking for a sequence and all. Just coordinates we require. Okay.
Mm.
Yeah. Then you need to be mentioned, report the total number of transcript and total number of genes. But this question is not as easy as the previously what we discussed. Like you need to count the number of rows, and for the genes you need to be remove the duplicates. Because whenever you are going to retrieve the data for all the genome, there are chances that you are getting unlocalized, you are getting the random and the alternative chromosomes. So first you need to remove that chromosome and then you need to count. This is what hint I'm giving to you. Okay? Is it clear?
Ma'am, can you repeat it again?
Uh, you in the whenever you are going to retrieve the data for whole genome, there are chances that you are getting the random alternative and unlocalized chromosomes. So first you have to delete that from your list and then you need to count the total number of transcript and the total number of genes. This is what a hint for the question. Okay, is it clear?
Yes, ma'am. Okay. Yeah.
Hmm. N- Now retrieve the third question. Retrieve the UTR region coordinates. Retrieve the UTR region coordinates in all transcript. Sorry, just a second. Now, for this, what you have to do, like you have a CDS uh, coordinate. You are not having the UTR coordinate there. And also, there are mixed strands, like a forward and reverse strand. So obviously, as per the forward and reverse strand, uh, the directions will be different, 5 prime and 3 prime, right? So first you need to separate all the strands like a forward one side, reverse one side, and then you need to calculate the UTR coordinates for all the CDS region from all the from the CDS regions, because there also you will have transcript start, you will have transcript end, you will have CDS start end, so you can easily calculate the UTR coordinate. It just you need to separate the data as per the strand first, because the directions are different as per the strand, right? Is it clear?
Yeah, ma'am.
So these are the three questions. You have a time till Monday. I will ask this on Monday. Okay. So I hope you all have set the requirement for WSL. Copy all this question, save in your system. Uh, Otherwise, like, yeah, just save in your system, and then what you have to do is you need to restart your system, and that's it for today. Then we'll discuss the WSL installation on Monday. Friday, the session will be for the Pymol, and tomorrow we have a holiday due to the Independence Day.
Okay. Thank you, ma'am.
Okay, you're welcome. So happy Independence Day to everyone. We'll meet again on Monday.
Yeah, ma'am.
Uh, Anyone like any doubt? Sonali ma'am, Sanya ma'am, Namrita ma'am.
No,
No, I
ma'am.
was just thinking that
Uh-huh.
you're saying to retrieve the UTR region of all transcripts.
Hmm.
So, like here we generally subtract uh, the regions, right? Because we know the
Right,
CDS, the UTR.
right.
So for
There. So in the
all the
first. Hmm.
so for
Yes.
all the transcripts, it will be a I mean, a very
Yeah, in
long.
Excel it is possible. You if you apply the formula for one, you can
Yeah.
just drag and drop it. That's it. Autofill it, right?
Yes, but the CDR regions will be different, right?
But if you will give the formula, if you
Yeah.
apply the formula and autofill it, it is going to be automatically applicable to all the rows, even if you
Yeah.
have a different CDS region.
Yeah. Okay.
Everything I told you, you just need to solve, right?
Yes.
Yeah. So not an issue. Just try and solve this. You will be able to do it.
I have a doubt.
Hmm?
Ma'am, in last class that we have done, the phylogenetic tree, actually I am getting a different result. Can I show you?
Okay. It's okay. Like maybe you have given some different options. So this can happen. Not an issue.
Ma'am, actually I'm not getting the original tree and bootstrap consequences tree tab.
Okay. So what I will suggest you once, just go through the recording. Just try it for the nucleotide sequence.
Yes, ma'am.
Okay?
Yes, ma'am.
Yeah. If you want to show me, that's fine. Like you can share your screen. But I think some part you missed, and that's why you're not getting the proper results.
So, ma'am, once again I will check and I will inform you.
Yeah, no problem. Take your time.
Thank you, ma'am.
Okay.
Thank you.
Okay then. Bye. Have a great day.
Same to you, ma'am.
You're welcome.